Hi, I'm Logan, one of the managers of MCAT content and curriculum here at Kaplan. In this video, Kevin Anderson, one of our expert MCAT instructors, will show you how he attacks some of the toughest discrete questions on the Bio-Biochem section of Kaplan's free MCAT practice test. This test was designed by Kaplan's question writers and validated by Kaplan's psychometrics team to be as representative of the actual MCAT as possible, so you know you're getting a great representation of what you will see on test day. Follow along to see how Kevin attacks these discrete questions using Kaplan's question strategy.
From the first few words in the question stem, it's obvious that this is talking about neuroscience. Specifically, this is asking for the effect when a normal component is missing or damaged. In other words, to answer this, we have to know what the function of an oligodendrocyte is so that we can say what will or will not occur if it's damaged. So I choose to rephrase this as what is the function of oligodendrocytes? Neuroglia, or simply glial cells, are a classification of cell that help provide structure to nerve cells and assist them in functioning efficiently. Glial cells that will be important to know for test day include astrocytes, microglia, ependymal cells, oligodendrocytes, and Schwann cells. Now, oligodendrocytes are the myelin-forming cells of the central nervous system. This myelin is a phospholipid substance that wraps itself around and insulates neuronal axons in order to allow for a faster method of signal transmission known as saltatory conduction. A patient with MS or multiple sclerosis, which is causing damage to these oligodendrocytes, is going to cause damage to the myelin sheath on the nerves in the central nervous system. Without myelin, we can predict that the signal transmission would occur slower than it would in a healthy neuron. All that's left to do is find the answer that matches this most closely. Now, B and D both mention a threshold, which would not be affected by the myelin on the neuron's axon since the signal threshold is reached at the soma. These are going to be out of scope. A says that healthy neurons dissipate their signal more rapidly. This is actually opposite since healthy neurons have a myelin to prevent the signal from being dissipated and help it to transmit through the axon. That leaves C as the correct answer, which explains what would happen when a neuron loses its myelin. Now, there's a few things in question 10 to point out that'll actually help lead to the correct answer. First, it's stated that the bacteria have lost the ability, meaning that bacteria are normally capable of this, and those genes are already present in its genome. They're just no longer being transcribed. And that's because the genes are being interrupted by these large DNA sequences. Next up, when the bacteria are put into a new environment, that function returns. In other words, the gene coding for surface adhesion is now being transcribed when before, while the bacteria were cultured in space, it was not. To me, this sounds like a form of gene regulation, but it's slightly more complex than that since the gene is being interrupted by DNA segments, almost as if a mutation occurred. If there's one thing that we don't want to do, though, it's overcomplicate the question. So I'll keep the rephrase simple and just stick to what I do know is happening. How can genes be transcribed in certain situations, but not in others? Now, the answer to this can be many, so process elimination is going to work best here. When a bacterial cell incorporates foreign genetic material from the environment and begins transcribing it, that process is known as transformation. This is a type of gene transfer in which the E. coli would be gaining new genes. This is not correct, though, since the question says that the genes for surface adhesion already existed in the E. coli genome. B mentions conjugation, another form of genetic transfer involving two bacterial cells and the transfer of a plasmid from one cell to the other. While there's no way to tell if this occurred or not, the second part of the answer doesn't make a whole lot of sense. Conjugation would incorporate new DNA into the genome, but it wouldn't restore the E. coli's normal gene function. C also describes a process of gene transfer, but recall that the E. coli in the question already contained the gene for surface adhesion, it's just been interrupted by other DNA segments. Gene transfer and incorporation of new DNA does not explain the simplified version of the questions as how are the genes situationally transcribed. That leaves us with D as the correct answer. But Why exactly? Well, there are certain segments of DNA that can move around the genome and integrate into different locations. These certain segments are known as transposons. They are found in both prokaryotic and eukaryotic cells, though there are no known active ones in the human genome. Essentially, transposons can cut and paste themselves into different parts of the genome, and if this occurs in the middle of a coding sequence, they can alter gene expression or cause mutations to occur. While this may not have been your first thought upon reading the question, it still sticks to the basics of variations in gene expression in different environments of genes that are already present in the genome. This is a convoluted way of asking a very simple question, and the hardest part is just sifting through the non essential information. If we get rid of most of this question, we can probably answer it more effectively, so let's do that now. This is what makes the rephrase step so vital. The question becomes much simpler when it's just asking, what happens to a repressible operon without its inducer? Again, using the methods to recognize patterns and simplify, a glance at the answer choices shows that we only need to know where the repressor binds to the operon. The second half of each answer choice is irrelevant. Using this method allows for a much more straightforward question. In fact, I am even inclined to change the rephrase to something even simpler. Where does the repressor bind to an operon? And that can only be in one location. The repressor binds to the operator, which blocks transcription of the operon. At first glance, the question seems more intimidating than it really is. While you are not expected to know exact values of gas pressures, you are expected to know the general principles of how gas exchange works. And those principles are pretty simple. Oxygen moves from the lungs into the bloodstream so it can be transported to other tissues. And gases move from a high to low pressure area. If the capillary pressure is 40, alveolar pressure must be greater than 40, and only one answer choice fits that description. All the other pressures are too low and would result in oxygen leaving the blood and entering the lungs. Questions on experimental technique are not uncommon. Most of these techniques you've either performed in your undergrad lab classes, or at the very least, you've learned about the process. However, most of us simply go through the motions to just get through the lab as fast as possible without really considering what we're doing in any step of that experiment. It'll benefit you greatly to take the time now to understand how and why these techniques work. Using a pH gradient to separate proteins does so based on the charge of the protein at that particular pH. The proteins are loaded into a center of a gel. Positively charged proteins will migrate to the negatively charged cathode, while negatively charged proteins will move towards the positively charged anode. As the protein moves through the pH gradient in the gel, it will eventually reach a part of the gel where the pH matches the isoelectric point of the protein. And at that point, the protein becomes neutral and stops moving since there's no charge to drive its migration through the electric field. This technique is known as isoelectric focusing. Questions which list multiple techniques are a great way to learn the differences between them during your review steps. SDS and reducing SDS page are similar techniques in that the SDS coats the protein in a uniform negative charge so that only their size affects the movement. Additionally, both gels have a consistent pH and not a gradient throughout. The reducing component reduces disulfide bonds and denatures that protein to its primary structure, taking the shape and folding of the protein out of the equation. Lastly, gel filtration chromatography is also known as size exclusion chromatography and uses a porous stationary phase. A buffer containing the sample is poured into a column. Larger molecules that are too large to enter the pores of the stationary phase will move through the column and elute first, while the smaller molecules are going to get stuck in the pores, allowing for them to be separated.